Good afternoon. So we are going ahead, in research methodology. So you all also have gone ahead. You have got iCards, you have got projects, and you will start research very soon, So research and public ethics we were discussing, lecture 8 and module B. So we did research methodology RM, rest part. MA madam will come and tell you the mathematics part. A lot of mathematics will be there in that. We will just introduce a term here today. So we completed what is this part, the green part in green. So we'll just, as in every day, we will just recap what we do in the previous classes, so that you घर पे जाके तो research methodology तो नहीं पढ़ेंगे, है ना? Definitely. So we will revise it here only. So there are research stages. By this time you should be aware there are 11 research stages. Okay, and we are going through one by one. But the initial stages are more important, because final stage mein tum already you are there. Then you are structuring is difficult. What you can do, you can improvise what you have already started. So like that, these are the stages, and I told you these are not that one is dealing from the other. They are completely linked. Suppose one analysis, one data collection is over, one analysis over, you have published one paper, 2 or 3 data collection is going on. Whatever you have learned from the first paper, you will try to improvise and implement it in the second and the third articles, right? And then whatever the HBNI doctoral committee, that means the experts in your field are telling, you will try to implement it day by day, and improve your research output. And also discussion with your guides, the lab mates, the lab which you join, you will definitely be not working alone, right? You will work with a team. And then better discussion over scientific problems where you are stuck up, like that. And so all these will help you. Discussion with the experts in doctoral committee, discussion with guide, co-guide, lab mates, experts in your field, like that. And slowly will improve the research output. And as you work towards, then you will become a guide slowly. And this is what the whole process is. So first process in defining the research problem. What you say, you want to do it on your own. So the research problem is that it's selecting and properly defining a research problem. It's a necessity. Necessary because a problem well defined is a problem half solved. This is what we understood. The technique again is similar, that we propose the problem first in a general way, a very broad way, with some errors also. It is okay. First, when you are doing it, when you are doing the research problem, define. So initial is the proposal in the general way, then understanding the nature of the problem. How will understand? By surveying the literature, discussion with the experts, okay, and then you rephrase the problem, and finally propose the working proposition. One means the problem which you will work on for your research. The same thing you applies to individual articles as well. You have to define a problem and you start working, and then you make a literature survey, you do the introduction part. Similarly, like a thesis means the whole research, the PhD career which you will go through, individual papers also you will have the same uh, methodology to be followed. But the thing is that in the whole thesis, you will have to interlink it according to the problem which you are proposing. You cannot deviate too much from the problem. So be careful while selecting the problem, and uh, that okay, I have to restrict myself. Okay, we understood that we cannot deviate. We have to lie within the scope. So the scope is defined in that. The survey of literature, then we do. Then purpose is that we understand the background. We understand the मेरे को भी याद आ गया, हाँ? We understand the background. It helps in the next stage, means research hypothesis. Then it helps in research design and what we are doing right now, and also structuring the reports, the individual articles, the patents, BRC newsletter. BRC has its own journal, huh? And then your thesis, whatever you will be writing. So that will we help, because if you do a proper literature survey, huh, you will be helped throughout the whole process. So benefits is that you become familiar with the subject, understand the theories involved in your studies, individual studies, evaluate and assess the papers or the already which has been published, and prevent duplication. You do not repeat the same thing which other people have done. Some sort of variation should be there, huh? If it is little or big, whatever it is. How to conduct a literature survey? You will go to primary and secondary sources, and how you will choose those, which are the sources you will go through, that is which are relevant to a topic, means topically relevant, credible, and recent literatures. Okay, and we also read what are the points to be kept in mind. We did study how we write a literature review, how to write it, and then we proposed a working hypothesis, huh? That is already done. We have defined our problem. We have done a literature review. We already proposed a working hypothesis. How it is that after extensive literature survey, researchers should propose a working hypothesis. That is a research hypothesis, which is a tentative assumption made in order to draw out and test its logical and empirical consequences. How we will do that? Again, similar thing: discussion with colleagues and experts, examination of the data and records which are present, review of similar studies, exploratory personal investigation depending upon the research types. Okay, we understood what is the difference between the research question and the research hypothesis. That research question is a starting point. If you do not have a question, you will not be able to propose a hypothesis. But from a research question, the difference is that the question is a starting point. Curiosity driven is question format, open ended. But you cannot do an open ended research. We have to close down, huh? And there is a proper question, means there is a option for test. So that's what you do. You test throughout your research. Each paper test it. And we started research. Analysis of data in a manner that but what it is that we have to basically plan look your piece, you have to build what will not get direction where to start with, huh? So you answer certain questions which are what, where, when, how much, and by what means concerning an inquiry or research study constitute a research design. So it will increase your efficiency. You already know what are the steps to be followed one after the another in sequence or parallel, whatever it is. You know the time management, huh? You have already defined the timeline. इसके अंदर मुझे ये complete करना है, huh? So you have a time management, minimizes bias and errors, huh? And then provides reliability and proper direction. And we discussed what are the characteristics of research design. And then finally we came into the 10 guiding questions, means what questions you have to ask during your research design. Means you have the problem, you have the literature survey, and then you have the hypothesis. So with the hypothesis, sit down and understand, ask what is the study about, huh? What is the study about? What is the study about? Is that relevant, huh? What is the study? Why is the study being made? Where will be the study be carried out, huh? If you have answers to all these, you have a proper research design, huh? What type of data is required? Where can the required data be found? What periods of time will the study include? what will be the sample design, what techniques of data collection will be used, how will the data be analyzed, and in what style will the report be prepared. You all know that you will start your problem. What way the report will be prepared, we discussed the HBNI thesis format as well. Huh? This is how the report you have to prepare. And if you have the answer to these questions, you prepare a design. You will have basically 4 components. Huh? One is the sampling design. So how many samples you will prepare? Okay. So mostly you will be concerned with sample preparation. Somebody in material science at least. Huh? Whether it's physics or chemistry. So the sample design deals with the method of selecting items to be observed for the given study. The sample can be human also for statistical research, or we discuss now with the example. So we'll go again. The observational design that relates to the conditions under which observations are to be made. The statistical design considering with the question of how many items are to be observed and how the information or data gathered are to be analyzed. And the operational design that deals with the techniques by which the procedures specified in the sampling, statistics, and observational design can be carried out. So basically 4 types of components will be there in your research design. Means how many samples are there, how the observation will be made, and the data collection will be made, the statistics of the data collection, the analysis, the operation, means the research methods, the techniques which you will use during your hypothesis testing. Okay, all these will come under these 4 categories. And what are the features of a good design? That the features are, it is the design should be flexible. Okay, it's not, should not be rigid. You have to keep options. Appropriate means topically relevant. Efficient means the minimum time, minimum cost. All these have to think. Economical, generally result which is minimize bias, huh? Well, not in one way. Okay, and maximize the reliability is a good design. The design which gives smallest experimental error. We understood what is experimental error with the help of variables. So we'll be basically dealing in each data collection, in every data, or in broad the whole thesis. There will be a relationship between 2 variables, independent variable and dependent variable. Okay, we have to minimize error means we have to minimize the influence of extraneous variable. Okay, we have to do design control experiments. So that we will come again. A design which yields maximum information and provides an opportunity for considering many different aspects of our problem is considered most appropriate and efficient design in respect of many research problems. So this is what we are the features of a good design. And then we understood the concepts. There are 10 concepts in research designs. Okay, for each hypothesis you have to understand that each of these concepts, what are the concepts? These are the variables, that is the dependent and independent variables. Then another variable comes into play, extraneous variable. Then there is a control, what it is. Then confounded relationship, research hypothesis which you have already proposed, experimental and non-experimental hypothesis testing research, experimental and control groups, treatment, experimental units. So this is what we defined in details yesterday. We will just see in short what it is. Just concentrate here. In short, this is not in detail, so you will not get bored here because this is given just in concise manner. Dependent and independent variables are, independent variable is what the researcher manipulates. Okay, independent variable में तुम changes लाओगे, huh? You will try to change the independent variable, and the consequence of that change you will measure in the dependent variable. You have to measure the dependent variable, the outcome. Okay, so this is the 2 variables you are dealing with, but there is a third variable you are not dealing with, but it has impact. You have to be careful during the design. The third variable is the extraneous variable, that is factors other than the independent variable that may influence the dependent variable if not controlled. If you are not doing a control experiment, extraneous variable will influence a dependent variable, means your measurement. Okay, extraneous variable, outside, something outside of your study, will affect your measurements, and you will have result in errors. Okay, so what you have to do, you have to do control measures to neutralize or standardize the extraneous variable. Okay, there will be no effect of extraneous variable. That type of experiment are told as control experiments. Confounded relationships occurs when an extraneous variable influences both IV and DV, both independent variable and dependent variable. If an extraneous variable affects, then it is said that we have a confounded relationship. Okay. Then research hypothesis, we did a complete class on it. That is a testable prediction linking the independent variable and dependent variable. That is your research hypothesis, which you have already proposed, right? Experimental, non-experimental hypothesis testing research means now you have the research hypothesis, you will do an experiment to test it, or you will not do an experiment, but you, with the help of observation, you will test it. So anyway, it is hypothesis testing either through experiments or without experiments. So these are the sixth component. Seven, these experimental and control groups. If you have two groups, you can have experimental groups and control groups. So in control group, you will do an you will not do any treatment, and in experimental group you will do a treatment. Or if you have a usual treatment, if both are having an effect, you will have a special effect in an experimental group and an usual effect in the control group. Okay, these are the terminologies, and try to understand what are these things. Treatments means the specific intervention or the manipulation you are giving to the experimental group. Okay, and experiment is a structure study which you will do, that is a treatment given to the experimental group. An experimental unit is nothing but the smallest unit of your experiment. Okay, so these are the 10 important terms. Again, I'm telling that a dependent and independent variables, two variable, that is their relationship is the research hypothesis. You manipulate the independent variable, measure the dependent variable. Extraneous variable is something out of your control which affects the dependent variable and creates error in your study. So you have to do control experiment where you neutralize or do not allow the extraneous variable to affect your dependent variable. So that all thing you have to keep in mind. Okay, and then if a confounded relationship is if an extraneous variable anyway comes, then there is a confounded relationship. And hypothesis, you have the, if you are testing the hypothesis, depending upon the research type, it can be an experimental hypothesis testing research or a non-experimental. Most of you will do experimental hypothesis testing research. And there are two types of groups, experimental and control groups. So experimental groups where you manipulate the independent variable and measure the dependent variable. Control group where you do not put any manipulation. Under usual conditions, you measure the dependent variable. Treatments is the specific manipulation you are giving. Okay, and experiment is a structured treatments, and experimental unit is the smallest unit which is in which the treatment is applied. These are the 10 concepts of research design. So we did with example. We repeat one example to understand that there is a hypothesis in sociology that more use of social media leads to lesser community participation. So how they will design their experiments just from nowhere, anybody can design, right? So how we will do? So there, the social media use and the community participation, these are the two variables. Okay, the independent variable is social media use, and on this the community participation is depending. So this is the dependent variable, the community participation. And extraneous variable means the education and age creates a factor, because a young person will do a social media use at a much faster and a more efficient way. So their effect on the community participation may be different from a aged person like me. Okay, so so the same experiment if they do on you and me, they will find different results. Okay, so that is the thing. The person who is designing the experiment, they have to take care that this is the extraneous variable will not allow it to affect my outcome. Means I will not allow that the dependence, this hypothesis outcome will become erroneous if we do not take care of the age of my group, okay, that I am selecting. So the control again is demographic matching, the same place it should be. Type is non-experimental. You are not manipulating anything. Okay, you are not asking, you are not teaching them anything that social medias are the use karo or something. And then we understand with the help of psychology, mindfulness practice, yes. Okay, so how we will prove it? With the help of research. Sociology, psychology student will do that. So they will train a group of people with mindfulness training, and they will keep another group placebo, means control group. There an experiment is taking place. An experimental group, they will manipulate. They will treat the mindfulness. They will what they will do? They will train them mindfulness, and they will see the outcome whether it is reducing stress or not. Okay, but there the extraneous variable, as I told you, the sleep quality also affects your stress level. So somebody having better sleep quality will have a different effect. So we have to ensure that the sleep quality on both groups are similar. Age is similar, profession is similar, because it has a direct impact on the stress. Like that, all those things are coming into play. So this is how it will look like, just with two examples. We did many examples yesterday, and then we go continue our research stages. That is preparing the research design. We are continuing, okay, because we are doing. Most of us are doing experiments, so we will discuss more on experimental designs. Okay, so we introduce more on experimental design. So as I told you, so important experimental designs are two types, in broad way: informal experimental designs, and formal experimental designs. So formal is more methodical, informal is more casual sort of thing, casual design you have to make based on your hypothesis. So in informal experimental design there are three types. They say the three types are before and after without control design, after only with control design, before and after with control design. These are the three types of experimental design: informal and formal. In the formal case, there are four types. That is completely randomized design. They call it in short CR design, randomized block design, RB design, Latin square design, LS design, and factorial design. So this is how you evolve the design, the experimental design, where you are concerned with experiment. Non-experimental hypothesis testing, these will be not concerned with. But since all of us, if not all, most of us will do experiments, okay, we will try to manipulate the independent variable of our study and find out the effect on the dependent variable. We have to understand what are the different types of design here and now. This. So before and after without control design, we do not have a control here. So in such a design, a single test group or area is selected. So here you select a single. So first you try to understand, then I will tell you an example. In such a design, a single test group or area is selected, and the dependent variable is measured before the introduction of the treatment. The treatment is then introduced, and the dependent variable is measured again after the treatment has been introduced. So before the treatment you measure, and after the treatment you measure, but there is a single experimental unit. The effect of the treatment would be equal to level of phenomena after the treatment minus the level of phenomena before the treatment. So there is a test area. Level of phenomena before treatment, you are before the treatment you measure the phenomena, okay, that is, you are telling X, okay. You introduce the treatment means, again, we studied just before what is treatment, okay. So we just studies that these are the terms, okay. So treatment is a specific intervention or condition applied to the experimental group, and the experimental group is one here only, okay. So you are giving a treatment, you are and phenomena means here they are talking about the variables, okay, which we are measuring, the dependent variables. So here what it is, they are telling that before the treatment the level of phenomena was X, and you manipulate something, you introduce a treatment, and after the treatment the level of phenomena becomes Y. So Y minus X is the treatment effect. So this is the basically the research design where you will find out the effect of treatment and measure it with the help of a single experimental unit. You measure your phenomena before the treatment and you measure the phenomena after the treatment, find out the difference, and you tell this is the experiment treatment effect, and you report it. Okay, so this is how a simple informal most simplest informal experimental design is. The main difficulty of such a design is that with the passage of time, considerable extraneous variation may be there in the treatment effect. But here they say in such a design, extraneous variable effect is more. Okay. As I told you, that these designs, experimental designs, were mostly introduced based on the concept of agriculture. Okay. Introduced during the fertilizers and all those means structuring. So what you are doing, research has been going on in scientific world from long back. But what UGC is asking, you structure your research from the initial stage. If you understand all the research methodology, we can structure my research, give a blueprint and then follow it, okay, according to the blueprint. So structured way people have done for mostly many types of research. Agriculture is one example, and many introductions were there based on agriculture. So this is we give an example for agriculture only here. So before and after without control design. So there is a plot. where we are cultivating something. Okay, and this is known as plot A. Okay, so the setup is that a farmer measures the wheat yield in his field before applying a new fertilizer, and again after applying it in the next season. So in one season, this season 2024, a farmer which is the owner of this plot, or he has rented that plot, whatever it is. So that in that plot he grows a crop, okay, and find out the yield. And next year he introduces a fertilizer in that, and again measures the yield in 2000 this year. So what will happen? The difference with the fertilizer, okay, whatever yield is there, say he has gone 25 tons, and without the fertilizer he has got only 15 tons. So he says, with introduction of the fertilizer, this fertilizer X, I have got an increase in 10 tons, which is a huge percentage wise of my product. Okay, so then other farmers will be inspired, and then they will also start introducing that fertilizer in their field. So this is a study which is of relevant in agriculture, and it will help in increasing the product yield, and it will help us also. Okay, so we are directly linked with farmers. So this is a example of simple informal design, most simplest informal design, that is before and after without control. So before there was no control, after there was no control. In the same plot they have done before measurement and after measurement. So before introduction of fertilizer, a measurement, and after introduction of fertilizer, a measurement in the same plot. Two different experiments in two different timelines they are done. But what is the problem is that limitation is, if yield improves, so this in 2025 yield has improved. It may be due to the fertilizer. Yeah, we agree that it may be due to the fertilizer, but could also be because of rainfall. So 2025 the rainfall is different, okay, than the previous year. Better soil moisture, the moisture content of the soil initially, which was there before in 2024, might have changed in 2024. Or seasonal effects, okay, they have not grown in the same season. So these are the extraneous variables. Rainfall, which he has no control over, okay. Soil moisture or season, okay, which he has not taken care of. So that can also have an effect, and you end up in a confounding relationship or confounding factors, which we have to avoid to get into a good design. So this is not a way of good design, but okay, we will agree. So ऐसा नहीं कि we will not do a simple study. We will do a simple study, and we will target some better study as well. Okay, these are this is a very simple experiment to do. As we complicate things, it will become more and more complicated. So no harm in doing, but you have to know the consequences. So a simple study you do and you tell I will publish in a very good journal, it will not be possible, right? So you have to accept what you are doing. So this farmer has to accept that okay, there were certain limitations in his study. Now we go into the second type, which is after only after the measure. They are measuring the dependent variable only after the experiment, not before. So what is this type of design? In this type of design, in this design, two groups or area are selected. Here you have two groups, okay. One is test group, test area, and another is control area. So two areas we are selecting. So in this design, two groups or area, test area and control area, are selected, and the treatment is introduced into the test area only. You have test area, okay, you have control area. We introduce the treatment in only the test area, means experiment you are doing, and here this is the control, okay. Control means no manipulation, usual conditions. In this treatment is introduced in test area, means experimental area where you are manipulating, and in both you are measuring the phenomena output, the dependent variable. So the dependent variable is measured in both the areas at the same time after the introduction of the treatment, not they have done initially. So after the introduction of the treatment, they have done it for Y. They have done it for the test area. They have done it for the control area. Treatment impact is assessed by subtracting the value of the dependent variable in the control area from its value in the test area. So where the treatment is introduced is Y, and where the control area, where treatment is not introduced, is Z. So Y minus Z gives you the treatment effect, different from the previous one, okay. Yeah, you have two types of areas, okay. One is experimental area where you are manipulating the independent variable, and one is independent variable means one of the variable which you will find proposed in the hypothesis, and on it, on the independent variable is dependent another variable which is dependent variable which you are measuring. So the effect you are finding out. So effect you can find out previously by only a single area before and after measurement. Here after only after the treatment introduced you are measuring. Okay, so this is the second type of design. The basic assumption in such a design is that the two areas are identical with respect to their behavior towards the phenomena considered. If this assumption is not true, there is a possibility of extraneous variation entering into the treatment effect. However, data can be collected in such a design without the introduction of problems with the passage of time. In this respect, design is superior to be before after control design. So before and after control design से ये better है. हाँ, जो previous design देखा इस ये better है. So this is what, in terms of agriculture, we do the same experiment. The farmer, a different farmer, what he does, he has two plots. He is a rich farmer. So he has plot A and plot B. Okay. So here in the two plots, what he does, he produces the grains in plot A with fertilizer, in plot B without fertilizer, and measures only the outcome, the yield. With fertilizer, he get again 25 kg, and without fertilizer, this time he gets the same 15 kg. If you do both the experiments, you can find out the conclusion. But here they are measuring only the outcome only in a single experiment. So setup is two plots are used. Plot A is given a fertilizer, plot B is left untreated. At harvest, the yields are compared. Limitations, since no pre-treatment measurement was taken. पहले कुछ नहीं किया था. Initial differences, soil fertility, pest population might bias the results. So here, rainfall will not be a factor. Okay, season will not be a factor because the same time, the time effect is reduced or removed from your measurement errors. Okay, because we are at the same time in 2025 itself, we started, the rich farmer started to have two plots and started the measurements. In one, he has applied fertilizer and measured the crop yield, and on the other, he has not applied the fertilizer and measured the crop yield. So, okay, and so here the extraneous variable is not rainfall or season. Here the extraneous variable is soil fertility. If his plot B was in Uttar Pradesh and plot A was in Tamil Nadu, we will have different type of outcomes, and he will end up in errors in telling about the product output with respect to the fertilizer. Okay, crop yield or fertilizer का जो hypothesis था वो errors के साथ आएगा. They will definitely get some thing, but there will be a confounded relations and it will have been errors. So the final informal design is introduced where such errors will not be there. So before also you will do and after also will do in the control design. Same, दोनों को combine कर दिया. Okay, simple. In this design, two areas are selected and the dependent variable is measured in both the areas for an identical time period before the treatment. Okay, before the treatment, उसने किया है. The treatment is then both introduced into the test area only, and the dependent variable is measured in both for an identical time period after introduction of the treatment. So that there is a test area, there is a control area. So level of phenomena before treatment, it is measured, and then the treatment is introduced, level of phenomena after it is measured. So what is this? This is y minus x. This is the test area results. Similarly, the control area where the level of treatment is A initially, and after that, without treatment is Z. Then Z minus A is the control. So if you have experiment minus control, what you get is the treatment effect. Here the treatment effect is determined subtracting the change in dependent variable in the control area from the change in dependent variable in the test area. So whatever the treatment effect in the test area, in this area, if you subtract whatever there is in the control area, this will yield you the treatment effect, and you can test your hypothesis, whatever you have proposed between the fertilizer and the product yield. This design is superior to the above two designs for the simple reason that it avoids extraneous variation and resulting from both. Previously, we removed the passage of time, but here the passage of time and non-competitive of the test and control areas, that will also be removed. Okay, so this is what, how did, so baseline yield before. So he has the two plots. He did in 2024 also the baseline yield, and for plot B also he measured the baseline yield. Now he does it for plot A in a new year with fertilizer, and in plot B also and without fertilizer. So setup is two similar plots are selected. Both plots baseline yield is measured. Plot A baseline yield is measured. Plot B baseline yield is measured. Plot A receives a fertilizer. Okay, and plot B without fertilizer. The plot is done. At harvest, both are measured again, and the change of yield is compared. Okay, with the control and with the experiment. The strength is that this gen is stronger because it controls for baseline differences in soil quality. Fertility is also introduced in addition to rainfall or seasonal effect. So all the parameters are controlled in such a design. So this is a type of experimental design which they say before and after with control. So here you can extend it to other examples, as which I try to do in every class, that before and after without control. Education, a teacher tests students' math score before introducing a teaching app and then retest them after a month of using it. Improvement could be due to app, but also due to practice, how much practice he has done or the time. Psychology also, a group of people stress levels are measured, then they undergo meditation workshop. Stress is measured again. Okay. The reduction may not be only from meditation but also from other lifestyle changes. Healthcare also, uh, patient's blood pressure is measured before a new dietary plan and after 3 months. Any improvement may also come from other lifestyle changes. So lifestyle changes say wo ho raha hai. So those are the extraneous variables which are affecting. After only with if you do the same things, two classes are taught with different methods. At the end of the term, exam results are compared without checking initial ability levels. Okay, then there will be errors. One group undergoes cognitive behavioral therapy, another doesn't. At the end, anxiety levels are compared. Health also, one is given a new drug and another is placebo. One on final recovery rates are compared only on final. But if you do before and after, then it is better. Two student groups are given a pre-test. Initially, you give a pre-test, right, and then select. Okay, both are having same average marks, and then you impart them two different types of educational program. The outcomes measured will be more appropriate than the other two, which we have studied. Same with psychology. Two groups of mentally depressed patients are assessed. One group gets a new therapy, and the other continues a standard treatment. After 6 months, both are reassessed. Difference in change indicates the therapy effect. With two groups have similar initial depression level, you can compare. But somebody who is having an initial level different, you cannot compare. So healthcare also, you can extend it such designs you can control. So what we conclude today is that before and after without control, easiest is very simple but weakest, no comparison group. After only with control, stronger, but initial difference may bias lesser. With time, biasness is removed. Okay, initial biasness. Huh? So we are telling the research design will remove bias. So this is how you can remove bias. If you have a design which have a before and after with control, the strongest of the three, as it accounts for both baseline means reliability, differences, and treatment effects. Okay, for biasness. So both are uh, compared. Okay, so this is how a simple experimental design, which is an informal design, is designed. Okay, so now we go on to the next one, which is the formal experimental design. So informal we have completed. Okay, three, and formal we'll just initiate CR design and then we'll do the rest in the next class. Okay, so but everything is given. So if one it will be shared. Still now it is shared? No, not they have they have not yet shared with you, huh? Only the online students are being shared. That means probably, huh? So formal experimental designs is we start with completely randomized design. It involves two principles: principle of replication and principle of randomization. You know what is this? Huh? Huh? You know? Huh? Yes. The We'll do it with the help of diagrams next class. Okay. The essential characteristics of the design is that subjects are randomly assigned to experimental treatments or vice versa. If we have 10 subjects, हमारे पास 10 subjects है, and if we wish to test 5 under treatment A and 5 under treatment B, the randomization process gives every possible group of 5 subjects selected from a set of 10 an equal opportunity of being assigned to treatment A and treatment B. One way analysis of variance. This is ANOVA. So this will be taught in the next classes, RM classes. ANOVA technique is used to analyze such a design. ये analysis ANOVA technique से होता है. It provides maximum number of degrees of freedom to the error. Such a design is generally used when experimental errors happen to be homogeneous. अगर सब experimental areas homogeneous into it's concerned with the field. Again, agricultural design. I'll come with the diagrams. It's missing here in the next class. So, if you're concerned with the field, then you will be ending up with if the field, all the fields are in Uttar Pradesh in the same area, or in Tamil Nadu in the same area, homogeneous, the soil qualities are homogeneous, then this type of design is appropriate. Okay, so इसके बारे में we will learn a lot in the next class. ठीक है? Today and tomorrow I have tomorrow also there is a class, two classes are there. So again tomorrow also I have to go a bit early, थोड़ा सा जल्दी, because there is a meeting. So for 1.5 hours we'll have class, and we'll go at a bit early tomorrow. So do you want me to start early, 2 o'clock? हाँ? कर सकते? Oh, you all are very cooperative. Like this is just a relationship I was establishing. No, till 1. You cannot complete lunch tomorrow, no? In that case. तो मैं तो